Welcome to my presentation. My name is Jiahui Cheng from Shanghai University of Sport. Today, I'm deeply honored to present our team's recent work titled Exploring the Therapeutic Potential of Sexual Cancer Based on Network Pharmacology. I would like to thank the organizing committee for giving me this opportunity, and I also want to express my gratitude to my supervisor, Associated Professor Bing Liu, for her continuous guidance. As we know, breast cancer is the most commonly diagnosed malignancy and the second leading cause of cancer-related deaths among women globally, with over 2.3 million new cases annually. Although targeted breakthroughs such as HER2 inhibitors, PARP inhibitors, and CDK4 and 6 inhibitors have significantly revolutionized clinical paradigms. Therapeutic resistance, severe hematologic toxicity, and poor blood-brain barrier penetration continue to compromise long-term clinical outcomes. Consequently, discovering novel anti-breast cancer scaffolds with innovative multi-target actions and lower toxicity remains a critical imperative in modern oncologic pharmacology. To address this challenge, our research group focused on a highly versatile chemical class, Thiazolone derivatives. Thiazolone derivatives are nitrogen sulfur heterocycles, recognized for their broad spectrum of biological activities, including anti-inflammatory, antibacterial, and anti-tumor properties. In our previous work, we successfully established a highly efficient one-step synthesis strategy utilizing a copper-catalyzed remote asymmetric propagation reaction. This method achieved high initial selective. This method achieved high initial selective coupling between propaglyl esters and cytolones, delivering target products with up to 89% yield and an exceptional enantiomeric ratio of 97.8 to 2.2. Thiazolone derivatives are nitrogen sulfur heterocycles recognized for their broad spectrum of biological activities, including anti-inflammatory, antibacterial, and anti-tumor properties. In our previous work, we successfully established a highly efficient one-step synthesis strategy utilizing a copper-catalyzed remote asymmetric propagation reaction. This method achieved high initial selective coupling between propaglyl ester and cytolones, delivering target products with up to 89% yield and an exceptional enantiomeric ratio of 97.8 to 2.2. Among the synthesized library compounds I'm emerged as our primary lead structure, as shown in the poster. Its unique architecture features a moderately flexible lipophilic isopropyl amino group linked via an acyl spacer to the Thiazolone framework. This design is intended to enhance structure complementarity with key binding pockets and improve favorable physicochemical properties. With this unique molecule in hand, we integrated computational systems biology to decode its mechanism before entering wildlife validation. To systematically uncover how compound I might fight breast cancer, we implemented a rigorous smart database network pharmacology pipeline. Structure descriptors were mapped using chemistry and processed via Swiss target prediction, target night, and superpride, generating 172 high-confidence unique target genes. Simultaneously, these targets were mined from gene cards and OMRM, uncovering 1,380 valid breast cancer genes. As illustrated by the Venn diagram, the intersection of these datasets revealed exactly 41 shared therapeutic target genes. We imported these genes into the strain database and escaped to map a topologic protein-protein interaction network by applying maximal lake centrality scoring. We selected the top 10 core hapgen. Next, we looked at the macro level by performing functional pathway enrichments, visualize the multi-dimensional mechanisms. We built an exhaustive component target pathway network. Gene ontology enrichment analysis indicated that these targets dominate critical biologic processes such as regulation of apoptosis, inflammatory responses, cell migration, and kinase-driven phosphorylation cascade. Currently, KGG pathway analysis mapped significant enrichment across 98 pathways. Most prominent include classical pathways in cancer, the PI3K AKT signaling pathway, and the PD-L1 expression and PD-1 checkpoint pathway. This suggests that compound I is ready to act via a multi-target mechanism rather than solely focusing on a signal protein, potentially surprising tumor survival, inhibiting metastatic migration and counteraction immune escape. Verify if compound I physically binds to these core proteins. We performed high-resolution molecular docking using AutoDock Vina. We simulated the binding orientations of compound I against our top-tier target crystal structures. The docking results for the top 5 proteins and compound I are shown in the figure, with the exception of NFKB1. Compound I showed favorable binding affinities for all tested targets. Specifically, it bound to JK2 with an affinity of minus 9.3 kilocalories per mole, stabilized by hydrophobic interaction with methylene 863, methylene 929, and leucine 983, alongside a salt rich with S959 for HSP 9081 at minus 8.5 kilocalories per mole. It participated in pi stacking with B133 and a T-shaped pi interaction with BIP157 for AKT1 at minus 8.2 kilocalories per mole. Its isobropyl amino acid side chain fits well inside the hydrophobic orbit, forming close contacts with leucine 156 and B161, anchored by an hydrogen bond with blue 234. Finally, we subjected these insights to absolute purification using vitro cell assays. To experimentally test our predictions, we performed the CK8 assays on compound A, B, and M in breast cancer cells. Within 10 to 200 micromolar, A and B showed little activity, while M exhibited moderate concentration dependent inhibition with an IC50 of approximately 96 micromolar. This modest activity might be partly explained by its flexible lipophilic side chain, which could enhance steric complementarity with target targets. However, we acknowledge severe critical limitations. IC15 near 100 micromolar is far from clinically relevant. Predicted multi-target mechanisms remain unvalidated and unvalidated molecular. We have not yet assessed the metabolic ability toxicity of target effects. Thus, we caution against over-interpretation. Our future efforts will prioritize two directions. First, mechanistic validation via Western blot and RT-qPCR to determine whether M genuinely affects the expression or phosphorylation of the predicted kinase. Second, the systematic structure optimization of M as a starting scaffold involving targeted modifications to its linker and peripheral rings with the goal of establishing clear structure-activity relationships and ultimately lowering the IC15 to an nanomolar or lower micromolar range. Given the preliminary nature of this study, there is substantial room for optimization. I highly welcome your critical feedback and suggestions to guide our future directions. Thank you very much for your attention.